So I'm interested in the fact that humans accumulate such a wide diversity and level of carotenoids in macular pigment. I was going to say a much greater fold difference in macular pigment across humans than the level of carotenoids across plants, but Antonio corrected my vision. I'm probably just ignorant of all the regulatory mechanisms that can modify plant levels. So suffice it to say that in many biologic beings, carotenoid diversity in terms of level is high. And understanding the factors that promote accumulation of macular pigments can help us have clues about the biologic systems that impact our ability to accumulate them, as well as give us information to maybe predict who might have higher and lower levels, and predict, as we've been talking about, people who may not respond as readily to oral intake. And ultimately, to identify people that might need larger amounts of carotenoids in the diet or smaller amounts of carotenoids in the diet to be both beneficial and safe. The data I'm going to talk about, as most of you are familiar with, from the Carotenoids and Age-Related Eye Disease study, a study of about 2,000 women between 55 and 84 that we conducted in 2001 through 4. We collected macular pigment density data with customized heterochromatic flicker photometry at several locations. I'm going to show you the data from 0.5 degrees. We selected that because there's the most between person variability and lowest within person variability at that location. But using some formulas provided by Nolan's group, we've also calculated macular pigment density area between those locations that we measured. And also from the formulas from Beakey, we were able to make estimates of dippers and ring profiles. And we've studied these in relation to determinants of macular pigment. And so far, we don't really come up with substantial differences from what I'm going to tell you today. So the data I show you will look at cross-sectional associations between macular pigment density and the intake of lutein from diet and supplements, a wide variety of other dietary and health lifestyles and eye history variables, as well as blood levels of lutein and other carotenoids available from analyses that Liz did, and genotyping for candidate genes for proteins that are implicated in influencing carotenoid flux from the gut to the retina. I'd also like to point out that we're happy that we will be able to see these women again, beginning even in January. So I'm hoping that we'll understand whether these determinants not only predict the levels of macular pigment, but perhaps the stability of macular pigment over time. And we're also going to have a chance to look at the relation between baseline carotenoids and the incidence of AMD, retinal aging, as indicated by OCT, as well as some of the visual functions. So I look forward to presenting that in future meetings. You've seen this slide many times that indicates macular pigment density levels by level of lutein and zeaxanthin from supplements and foods. And there's a linear increase in the average macular pigment density across intakes up to about 2 milligrams per day. The American average, sadly, I realized after listening to Antonio's talk, much higher in many other countries. But after that level, there's a small increase, but it largely plateaus with considerable variability around these means. We know previous trials of supplementation indicate variable response, with between 5 and 50% people in studies done between 6 months and 2 years who don't show any marked response at all. And we think that perhaps our data, the data points here, people who have very high intake but low levels of macular pigment are potential low responders, whereas people who have very high levels of macular pigment despite low levels in the diet are potential high responders. So I'd like to consider some of the influences on macular pigment levels, starting with the diet and the gut and moving on from that. So I'm using Patrick Borrell's slide a little bit modified to add some of our data. We first found and published in 2006 that a variety of dietary factors are related to level of macular pigment. One strong association was with dietary fiber, even after accounting for the carotenoid composition. This is similar to other absorption studies that have found that women who eat more vegetables and fruits have higher blood carotenoids even after accounting for the carotenoid presence. And so there are other factors, probably in fiber-containing foods, that are responsible for this effect. I've just started a collaboration with some people on the microbiome, where I feel like a kindergartners, we know so little, but also I've learned that certain fiber types are foods for healthier bacteria. So it's even possible that fiber in our diet can affect our microbiome populations and have health effects that we don't even know about. But there's enough complexity already to consider at one time. We know from other feeding studies, that the level of dietary fat matters. Also in our study, polyunsaturates and omega-3 saturates were associated with higher macular pigment density. Then Patrick Borrell has summarized the many proteins that are involved with absorbing carotenoids in the diet. And I've indicated in red stars by those for which we have found polymorphisms in the genes which encode those proteins. So this involves, these are all proteins which transport cholesterol and other lipids from the intestinal mucosa into the cell, usually in collaboration with transport of lipids and cholesterol, including SRB1, CD36, Niemann-Pick, and ABCG5 variants related to macular pigment density in our studies. We also found that variants in BCM01 are associated with macular pigment density, which cleaves carotenoids, although interestingly, lutein and zeaxanthin aren't particular substrates. And if there's time, I'd like to talk about a new idea that George made me think about for a reason why carotenoid cleavage enzymes might have an impact on levels of macular pigments that I hadn't previously thought about looking at it evolutionarily. We also found, but I see it's not on this slide, that on the plasma membrane side here, there are also transport proteins which influence, variability of which influence the level of carotenoids in the retina. Specifically, a large impact by ABCA1 transport, which encodes a protein, an apoprotein, on HDLs that's important for its stability. So this slide is just a summary of all of the genes that we assayed, that we genotyped, that many of you and others in the literature have suggested might be related to the intestinal trafficking of lutein and zeaxanthin in the gut, the blood, of the retina, cleavage, long-chain fatty acid status, and binding proteins in the retina, as well as retinopathy-related genes. These could all affect the amount we see in the macula. So in addition to those that I mentioned from the last slide that influence intake into the gut, there are others that influence intake or transport in the blood, such as the ABCA1. I need to step back just a second and tell you that I've indicated here when we found they've been associated with macular pigment levels and when they've been associated with serum levels indicated by an M and an S. So you can see many of the proteins I just talked about in the last slide are associated with both higher level, there are the variants, certain polymorphisms are associated with higher levels in the serum, as well as the macula, suggesting that it's an absorptive factor that influences their levels in macula. Whereas some are only associated with variants in one, some are only associated with values in the macula, or some only associated with variants in the serum, at least in these initial analyses of independent predictors. So many of the lipids that traffic carotenoids into the gut also traffic carotenoids into the retina and also throughout the retina. And we'll discuss these, which may play a role. We found no associations with the dioxygenase cleavage enzyme that are associated with macular pigment density. And we thought that we might see an association between long-chain fatty acid status and uptake of lutein and zeaxanthin, but we didn't see that at all. We do see that long-chain omega-3 fatty acid status, at least the genetic variants that predicted, are associated with macular pigment, both elongases and desaturases. So we think that probably this reflects something that happens in the retina. So the presence of EPA and DHA in the retina might stabilize lutein and zeaxanthin. We found, you know, that Paul has identified the zeaxanthin binding protein to be GSTP1, and indeed we did find that variant polymorphisms in this were associated with macular pigment. But we didn't see variations in the lutein binding protein to be associated with macular pigment. Instead, we found those to be associated with serum levels of lutein and zeaxanthin. 13 variants were. So this threw us for a loop a little bit, and this story will play out later. And then, as you'd expect, if the retinal integrity is lost, the ability to hold on to these carotenoids might be compromised. And indeed, we found that certain retinopathies are associated with lower macular pigment, although sometimes it was also related to macular pigment in the serum, such as this cleavage enzyme. So there may be multiple mechanisms happening at once. So these reflect the independent contributions. So oftentimes you see the results of ordinary least squares regression analyses that search for the high signal amongst the noise. What are the independent predictors? Oh, I got ahead of myself. I also want to point out that Chris Hammond's twin studies have shown us that they did a supplementation in about 300 twins, and they also found that women who had these variants were more likely to show a response to supplementation in either the serum or the macula, indicating that these epidemiologic data can provide important clues that might be borne out in clinical studies. So overall, looking at independent predictors, it looks like there are a lot of contributors, including aspects of diet, proteins that influence absorption and transport, and influence the uptake and maintenance in the retina. Also, what I haven't talked about, but I've shown in several publications, are many phenotypes are all actually related independently to macular pigment density, and most specifically, being obese or having diabetes is associated with lower macular pigment at all levels of intake, and we found several other phenotypes associated with that as well. But all of these things, from the massive data available to us from the Women's Health Initiative and CAREDS, we've only been able to explain 16% of the variability. Is there more? There's some minor players that we might be missing. And this was the purpose of the next analyses that we did. And I wanted to say that also some of these phenotypes may suggest influences on metabolic syndrome, which could affect the distribution of carotenoids throughout the body between liver, adipose tissue, and the eye. They could be differentially distributed depending on these types of phenotypes. But also it could affect the degradative processes of inflammation, oxidative stress, which could turn over these carotenoids at a faster rate. So because we can only explain 16% of the variability, but Chris Hammond's studies suggest that there's much higher heritability of macular pigment level that could be explained from the twin studies, and also from supplementation studies. We've used an approach to look beyond independently determined estimates, trying to model the complex and interacting factors that we know and that we may not know. We considered several models, and we settled on partial least squares regression modeling, which will identify the variables not only that stand out, but are team players, conditionally important only when other team players are around. And so this technique that Pat Borel has actually used, partial least squares regression modeling, seemed to be an interesting approach to try. How we went about this is we started with a lot of data. We had over 400 SNPs from genes, tagged SNPs in about 26 genes, 28 different phenotypes that I've overviewed so far. And we put them into the black box of PLS modeling. And the output gives you several possible models of the way these things fit together, from one latent component up to 30. And the first job is to pick the model that seems to work best. And our objective criteria for doing that involving the model that gives you the highest percentage of variability explained. So we chose the model with 3 latent components that seem to predict the most variability. And then we put it through rigorous leave one out cross-validation, taking people out one at a time and seeing if the estimates change much. And then when those 2 criteria are passed, you end up with a model. And this model contained 46 variables. And you don't know how they're interacting. It's a data-driven process, so it's not based on our prior hypotheses, except for the things that went into the model. And then, in order to make predictive measures, we used the multivariate regression modeling with backward selection to prune this to models that we can use for prediction, which I'll show you. Okay, the next 3 slides are busy. I'm going to first show you all the phenotypes in the model, and then in the next 2 slides, I'm going to show you the genotypes in the model. And certainly the independent predictors that I've told you about before were heavy players in this model, but a few new things have been found. So the output for PLS gives you a VIP score, or variable importance in the projections score, which give you an idea of how heavy in this model these variables were weighted. And you can see that serum levels of lutein and zeaxanthin were given the highest weighting, and also high weights for the lutein intake from foods, from fiber intake, as well as waist circumference. But the new players in terms of phenotypes were some that were associated with higher macular pigment and some with lower. We found that physical activity was associated with higher macular pigment in this model, as well as having arthritis, which threw me for a loop. I would have expected the other, but perhaps it's the large amounts of anti-inflammatories that people take when they have arthritis that might lower inflammation and degradation of carotenoids. I'll stop speculating, I'll never get through this, but the other players that were associated with lower macular pigment were retinol, interestingly, hypertension, which we expected, age, which some people have shown, and use of cholesterol-lowering medications, which also there's some evidence for in the literature. And some of those made it into the predictive model that I'm going to show you. These are all still part of the model, but if we're going to try to predict level of macular pigment, these are the values used. Okay, now there's 2 slides for genotypes, and what I want to highlight here mostly is that these are the new variables that popped up. So we found new evidence for SCARP2, which I know little about. Also for the name and pick see, like one gene, variants were associated with serum levels of macular pigment, or serum levels of carotenoids, but not macular pigment. But here we see an association with macular pigment. Also, this is a good example of being a team player. ABC G5 and G8 form a dimer in the intestine on the mucosal side that participate in cholesterol and carotenoid uptake. And while we saw in independent analysis ABC-G5, it wasn't until we put all the team players together that we also saw this other variance in this other gene explains some variability. But the other variables that you would expect from the independent models were still there. Heavy hits from SCARB1 and some hits from ABCA1. Then moving on to gene variants related to carotenoid cleavage. They were there as we expected from independent models. We saw two new gene variants that are associated with long-chain omega-3 fatty acid status, another elongase gene, and another desaturate gene, which is the rate-limiting step for EPA synthesis. Also, and here was our surprise, our main surprise, we found not the zeaxanthin binding protein, GSTP1, as we did in independent analysis, but rather the lutein binding protein, STARD3. Several variants in several SNPs were associated with macular pigment level. We also found that GSTM1 was associated with macular pigment level. Just a few more comments on these new ones that were picked out. I mentioned the ABC8 gene, which encodes the intestinal transmembrane transporter protein that forms the heterodimer. And so this was one novel finding. And also the elongase and the desaturase enzymes, which likely predict the type and amount of long-chain omega-3 fatty acids present in the retina. And STARD3 was the interesting finding. This is a lipid transfer domain containing 3 genes that's localized to many inner and outer retinal layers. I mentioned 13 SNPs were related, though, to levels of lutein and zeaxanthin in the serum, so I'm not quite sure how this fits into the puzzle of things. Maybe we'll figure that out at some point. And so what is it doing there? Well, STARD3 specifically in many tissues, placental tissue, adrenals, and brain, have been shown to initiate sterol synthesis by trafficking cholesterol into mitochondrial membranes where that process can begin. So it's a regulatory gene. And so we don't know exactly how this all happens or why this might be relevant in the retina, but I look to Paul for some possible ideas or perhaps data in the future to help explain this. And then we found GSTM1 encoding a glutathione S transferase. Don't know specifically why it's associated, but it is important in oxidative protection, so it might be that role which has supported this finding. So here we are. We've done all these analyses, entered in new players, but we can only explain 3% more of the variability in macular pigment levels. Nonetheless, some identified could be important cues to the physiologic processes that might be important. Also, it allowed us to develop a model which could allow people who don't have macular pigment data to estimate a person's macular pigment density level crudely. We found that using this model of all the players similarly classified 67, about 2/3 of participants, within a quintile of their actual macular pigment density levels. And it had a very low rate of misclassification. So, for example, women, the lowest quintile were misclassified into the 4th or 5th quintiles only 1 or 2% of the time. So it's not wonderful, it won't give an accurate estimate, but it would give you a crude estimate of somebody's macular pigment density, for example, in large epidemiologic studies that can't go back and measure macular pigment. So then, finally, we looked at the joint predictors of response to macular pigment, not actual response, but here we're using a theoretical model where we looked at people's level of macular pigment density and level of intake from the diet. And for these analyses, I'm only going to focus on women who have above 1.7 milligrams per day and higher. And the question we're asking is, what are the characteristics of women in this group who have macular pigment levels more than 2 quintiles below their level in the diet as potential low responders? The steps that I won't go, oh, I see I'm over, so I'm not going to go on and explain, but similar steps that were used for the other. The model included about half as many variables and was pretty predictive, having a receiver operating characteristic of 75%. Heavy players remained the same, so that if women had high waist circumference or serum lutein, they were more likely to be low responders. No, I'm sorry. If women had the abdominal adiposity in the largest tertile or diabetes, they're 2 to 3 times more likely to be low responders, whereas if they had serum lutein and dietary fat in the high tertile, they were 2 times less likely to be low responders. There are many genes that were associated, and I had made some modifications adding SCARB. So there were also 2 SCARB that I had not on the slide, but the modifications didn't come through today. So this should show the new players were CD36 as well as SCARB. And these are some pretty strong odds ratios when you consider that these are odds ratios for being a low responder for every risk allele. So if you have 2 risk alleles, it can get quite high, especially when you add them all up and put them in a genetic response score. So we found that putting people in quintiles for this genetic response score, considering all the phenotypes we talked about, new and previously discovered, and the genotypes, women in the highest quintile had a 30-fold higher chance of being a low responder with scores that were in this high quintile. So this could be used by, before you conduct a clinical trial, to identify people who are likely to be low responders and perhaps don't select them for short-term supplementation studies. It could be used post-hoc, such as an ARIDS2, where you can look at whether the hypothesized association is seen after removing people who are potential low responders. I'm going to skip that, which it basically says that the model is not substantively altered, but has more players. And the new players added are indicated on this slide. So in summary, it's identified some novel genotypes, which really require further testing. This is in women. We've been hearing this morning that response might be different in men and women and different in people with different ancestries. But you might be able to use this as a crude marker to estimate person's macular pigment level. And the empirical prediction model for low macular pigment requires further testing as well, but might be useful in clinical trials either that have been conducted or that need to be conducted to indicate low responders. So thank you for listening to me, although I've gone over, and I thank the contributions of the many collaborators as well as the funders. Thank you.
Thank you, Julie. And although Julie was right, her talk was a little long, I think we have time for 2 questions. Well, I thought maybe people were getting hungry there.
Thank you very much for a very interesting talk. Low responders are going to have a problem then? the low responders will have a problem. Is there anything about the information that you've uncovered from the gene profiles that might suggest a co-medication that you could then use that might support improved uptake?
Well, maybe Eric would have something to say about that. Medications to improve response based on things like ABCA1.
As far as I know, there's no real drug which would increase specific ABCA1.
Okay. So I
We're
don't
working
know,
on
but
that.
that is a good thing to think about. Yeah.
But there are many drugs which decrease ABC1 activity and many pathologies like hypertension and so on.
Okay. Over here.
Liz,
Liz?
I'll leave Liz,
Liz?
take the floor. I've said enough.
Hi, Julie. Your data is great. It's exciting to have this tool. But these are done in women. Do you have any reason to believe that it wouldn't or would work in men?
I think that some of the experimental data that I saw this morning suggests that women are more likely to respond. Certainly, these are small studies, and so we don't know. So I would really like to see, we're happy to give people our SNPs so if they've got studies in men, to see if they can replicate them as well.
Time for one last point.
Well, since you're an organizer.
No, I think it's important we kind of understand when we say responders versus non-responders. I'm not sure. I think Randy Hammett might have a view on this, but I think people always respond at a certain level. So I think how we define the type of response. I mean, what you're talking about is normal dietary habits, and then let's extend that to taking a supplement. How do people respond to diet or to supplementation by modifying something? And I've seen basically if you give enough of the stuff, you get them to respond. In the blood, pretty much straight away. I've only seen one subject in my recent clinical trial that showed minimal, if no response in the blood, and I'm not sure if that's purely down to compliance. So I think it's important. My point is, I think when we talk about non-responders, we're talking about in a normal kind of non-interventional world. But when you extend that into the interventional world, one big predictor of macular pigment is taking stuff in high concentration, taking the carotenoids in high concentration. So
Yeah, I appreciate that point of view, and I do think that we're not talking about non-responders here. We're talking about low responders potentially who might need higher doses, and this will be important as we move forward to looking at optimal levels. It might be that people with these characteristics need more. It might be people who are high responders need less.
yeah, for sure.
Well, let's thank Julie one more time.